So it gives me great pleasure to introduce Dr. Donald Karcher. He is currently the president of the College of American Pathology, and organization is dedicated to the best practices in pathology and laboratory medicine. He is a professor of pathology at George Washington University in Washington, D.C. He's been head of hematopathology there, and he's also the immediate past chair of pathology, a position that he held for 20 years. Dr. Karcher has had numerous leadership positions in national and international pathology organizations, but I think throughout his career has been characterized by steadfast advocacy for pathology and laboratory medicine. Most recently, Dr. Karcher testified at the congressional hearings on federal oversight of laboratory-developed tests on behalf of the good guys. So thank you very much, Dr. Karcher.
So thank you, Dr. Abruzzo, for that great invitation or introduction, and thank you for the invitation to speak before this group. So let me just give you a heads up. I'm not going to tell you anything you don't already know, so please relax, put your pens down. What I am going to try to do, and you can see the topic here, is to talk about federal regulations as they affect molecular diagnostic testing, which will include some discussion of gene patents. What I am going to try to do is to put this into perspective for you with a particular emphasis on how this seems to be changing, and it is changing. It's evolving, as you well know. If you followed the LDT saga, which, by the way, I will describe for you, you know how that went through all kinds of twists and turns. Well, regulation in general is undergoing those same twists and turns, and I'm going to try to give you a little bit of an insight as to how things change, things to look out for, how the CAP and many other organizations, including, I'm sure, this organization, are monitoring these things. I actually even have some breaking news that just happened here about 20 minutes ago, if you saw me getting up and in and out of the room. I'll share with you in a few minutes why I was out of the room. And then finish off maybe with sort of a resurgence of this whole issue of gene patents, and where that seems to be headed, and hopefully how we can make the right thing happen with that. As Dr. Abruzzo mentioned, I am a hematopathologist, so let me thank you for everything that you do for our patients. I use the data that you generate essentially daily. So I have no disclosures, and the learning objectives, they're a little wordy, but ultimately I'm going to try to do what I just said a minute ago. Try to put all of this regulation into perspective, talk about how we are constantly trying to balance innovation with patient safety, and how things are, how they look over the horizon going forward. So let's talk about just overall federal regulation of molecular diagnostics. So this is no surprise to you. Molecular laboratory testing is regulated under CLIA through CMS. That you already know. And as you also know, I hope, CLIA, which was passed into law in 1988, has not been revised legislatively. It has, however, been updated many, many times through the rulemaking process, and that continues through this day, often working through CLIAC. You probably know what I'm going to say about CLIAC in a few minutes, but that's been a useful methodology, maybe a little slow, but a useful way to update CLIA. New technology, and certainly the new technology NGS, but also what we just heard about. I had no previous knowledge of optical genome mapping. New technology such as that, and anything to do with AI, has and will continue to require regulatory oversight considerations, and we're all sort of learning how that oversight will be developed. Laboratory-developed tests are a different animal, and I'm going to spend a few minutes just sort of bringing us through the saga and where we are right now. So, as you're not going to be surprised to know, there's almost nothing said about molecular analysis in CLIA. By the way, if you've ever read the CLIA statute, it's 13 pages long. I have a copy in my backpack. I just keep it with me just in case. It's not very detailed, and all of the details that go into what we all do and how our work is regulated has been done by rulemaking, and the rules go on for hundreds of pages, but the actual statute is only 13 pages long. So these updates are really essential, and in the work that you all do, arguably some of the most necessary changes have to do with molecular analysis and genomic analysis. So that's what we have been working on in terms of updating the regs, and how, with a little bit more challenge now, we're going to continue to work with CMS on updating those regulations. Now, here's an example of how these things work. In 2020, CDC and CMS released a request for information regarding some changes that they perceived needed to be made that have to do with NGS and informatics and bioinformatics and storage requirements, data storage requirements, et cetera, et cetera. This is how these things happen. In 2023, they formed an NGS work group to determine the potential regulatory changes that might be necessary. Last November, that work group presented its findings, its recommendations to CLIAC, and they were approved by CLIAC. But you probably heard that the Trump administration disbanded CLIAC in March. So this makes our challenge even greater now. I'm not here to talk politics, but there were some potential really good things about the Trump administration's approach, maybe deregulation of a lot of things that maybe are excessive. However, I'm afraid to say that, from our perspective at least, it has gone way too far, and a lot of very important advisory committees, such as CLIAC, have been disbanded, and that continues to this day. So we are now challenged even more to work with CMS in a different format, and we are already working with them in a different format beyond CLIAC. Now, a lot of the regulations or the changes in the regulations are reflected in the checklist. If you're a CAP-accredited laboratory, you know about this. If you're not, and I'm not here plugging CAP accreditation, but just so you'll know, the molecular pathology checklist is constantly updated. Every year, we update everything in there based on new regulations, and those, by the way, have to be approved by CMS. We have deemed status from CMS to accredit laboratories. CMS has to approve the changes that we are reflecting there. Now, I'm not going to go into any detail. This is actually the table of contents from the latest version of this checklist that was just released last December. And you can see, just reading through the table of contents, the level of detail that this goes in. Checklist items have a very clear mark as to whether or not they are new regulations or revised regulations. And most of the things that are changed are revisions. And those revisions are gone over in great detail. So this is at least one source of staying completely up to date in terms of what the new regulations are and what they mean in the laboratory. A variety of the things that you do every day are listed here. The validation methodologies. We just heard about some exciting new technology that has to be validated, of course, to be used clinically. Just going through some of the things that you know well, several sections on NGS here, and the new regs as they relate to NGS. By the way, those bioinformatics changes have not yet been adopted by CMS because of the disbanding of CLIAC, so we're hopeful that those new requirements can be put into place soon. On and on. All the way at the bottom, personnel standards definitely need to change in terms of genomic analysis and next-generation sequence analysis. And so those changes have been reflected and will be reflected in the checklist. So the FDA's role here, as you know, molecular analysis, genomic and other molecular analysis, are considered medical devices by the FDA. And that the process whereby all of these things are approved go through either the pre-market approval process, which is for the highest risk tests. That's a much more detailed process than the so-called 510K clearance process, which is used for tests that are substantially similar to tests that already exist. But they all have to be either approved or cleared by the FDA first before they can be marketed. This is a complicated process. There's also the need to use the so-called quality system, meet the quality system requirements. All of this is very complicated. This is why all of our laboratories have had to develop laboratory-developed tests, because it takes companies a long time to go through this process. New variants are discovered. It might take 2 or 3 years for a new variant to show up in an FDA-approved test. All of our laboratories over the years have had to develop laboratory-developed tests. That's a real problem, as you know. It continues to be a problem. By the way, I'm happy to say that the FDA is somewhat flexible in their regulations. And the print's a little small, but there's an actual quote here from an FDA guidance saying that they're acknowledging that NGS can examine millions of DNA variants and thus warrants a flexible approach to oversight that is adapted to the novel and evolving nature of these tests. So it's not all black and white. They understand that some of the testing that you do, much of the testing that you do, requires a flexible approach. So I'm going to talk a little bit about AI, because as you know, AI and machine learning is entering into pretty much everything that we do, and that certainly includes the technology that you're using. The FDA, by the way, has been approving AI software as a medical device since 1995. 30 years they've been doing this. So this is not as new as it may have seemed. And as of last December, the latest report from 2024, over 1,000 devices have been approved by the FDA. All of these are static. These are not the algorithms that continue to learn. Regulation of the non-static or dynamic platforms or algorithms, which include generative and autonomous AI platforms, represent a tremendously higher level of difficulty in regulating, because they continue to change, and the regulation is going to have to figure out how to manage that change and to make sure that those algorithms are not drifting off or hallucinating or coming up with incorrect conclusions. So in 2021, the FDA released its AI and machine learning-based software as a medical device action plan, which includes a total product lifecycle approach to this regulation. Now, this is still taking shape. And here's the breaking news. As I was sitting in the front of the room here, a meeting with the FDA commissioner, Dr. Macri, was just confirmed, and I had to leave the room to indicate that I will be able to make that meeting on the date that it's scheduled. We're going to be talking to the FDA commissioner about how we feel they need to guide this guidance and guide this regulation in the coming years. They need to work with organizations like the CAP and many other organizations to make sure that they're hearing from the right stakeholders as this develops. They can't just listen to industry. Nothing wrong with industry, but they have to also listen to the people that are using the technology, and also and how it affects patient care. So this is just some recent regulation of AI that the FDA has released, one of which, the first one there, it's a guidance on clinical decision support software. That was released in 2022, and you might not think this has anything to do with molecular analysis, but let me bold one statement here, which is that clinical decision support functions that are related to NGS are cited in this guidance as not being excluded from this regulation. Certain clinical decision support software functions are excluded from regulation, but those that are at a higher level are providing more significant data for the patient are not excluded, and NGS is one of those. And then a couple of these other ones just deal with this total product life cycle approach to this regulation, because these tools will evolve, and the regulation has to reflect that and to guard patient safety as these algorithms continue to change over time. So it's not just about the government. I'm happy to say that the CAP is a founding partner of an organization called the Coalition for Health AI. And this is a group of stakeholders across the whole spectrum, from the health sector, including industry, government, academia, and patient organizations, that has been formed to try to make sure that the development of all AI in medicine is done responsibly, that it's deployed and its oversight is done properly so that we have safe, effective, and ethical AI tools to use for our patients. So I promised to tell you a little bit about laboratory-developed tests. You're all probably familiar with, the whole saga that we've been through with this. And it's been a very active issue for 15 years, and for better or for worse, I've been directly involved in it for all 15 of those years. So, and it's been quite a ride. The issue's actually been with us since 1938. I'm happy to say I was not involved in it back then. But it's been through all kinds of twists and turns and changes, and including some very recent changes, which I'll share with you. The recent FDA final rule was overturned by a federal court, as you well know. However, and it would be nice to say, okay, that's done. It's not. This is not yet fully resolved, and in the end, I think everyone agrees Congress has to act to sort of sort this out in the right way. So here's a very long list of all those twists and turns. It actually started with the Federal Food, Drug, and Cosmetic Act in 1938. More importantly, the Medical Device Amendments of 1976 is what the FDA has based its feeling that it has the authority to oversee LDTs. However, as you know, they practice what they call enforcement discretion over LDTs for decades, feeling that LDTs were simple tests developed in a single laboratory, not complex. Well, as you well know, because your testing is incredibly complex, many LDTs, starting about 20 years ago, started getting way more complex. And they recognized in 2010, they first announced that they were going to formally regulate laboratory-developed tests. They released their first draft guidance in 2014. That was debated for 2 years, and it was getting ready to be released, by the way, the final guidance. But a somewhat unexpected event happened in November of 2016. Donald Trump was elected president, and that stopped everything in its tracks. Now, you may be surprised to know that the Trump administration, the first Trump administration, felt very strongly that there needed to be legislation to sort this out. And frankly, as best as we can tell, they still believe that in the second Trump administration. Legislation was released. DIA, many of you may remember, the Diagnostic Accuracy and Innovation Act, which would have completely stopped accuracy, well, maybe not accuracy, but innovation in its tracks. We and pretty much every other organization fought that legislation, basically to stop it as best we could. The FDA also recognized that it was not the right approach. They did what's called a technical assistance. Federal agencies, when there's new legislation proposed, they actually comment formally about that legislation, how it might impact that agency, and they basically said, this won't work, here's another structure that we think would work, and that ultimately led to the VALID Act. Now, some in this room, maybe most in this room, we're not big fans of the VALID Act. I'll tell you, sort of a little bit background information about that. So, the VALID Act draft was released in 2018 by Representatives Bouchon and Digette. Representative Bouchon is a physician. There was a lot of debate, and stakeholders, including the CAP, we tried to make the VALID Act as good as it could possibly be. We were not big fans either. At the last minute, the Senate leadership asked us if we would support it, and we said, it probably has barely enough guardrails around what the FDA can and can't do that we felt like it's better than what the FDA might do on their own. I think in retrospect we were right about that, but that's sort of water under the bridge. Well, that law did not pass. The VALID Act did not become law. And the FDA made very clear in 2023, If there's no legislation, then we're going to regulate these tests based on our existing authority, statutory authority from that medical device, those medical device amendments of 1976. Their proposed rule was released in October of 2023. There was extensive input from stakeholders. As was mentioned by Dr. Abruzzo, there was a House committee, E&C committee, that was convened, and I had the distinction to testify before that group. Now, just to remind you, this rule that was proposed by the FDA would have exempted existing laboratory-developed tests from the 4 and 5 stages 4 and 5. This law would have rolled out over 4 years, but it would hold even existing LDTs to stages 1, 2, and most of 3. And we felt that this was absolutely not workable. Laboratories would essentially all have to stop developing LDTs if they had to comply with this. Now, starting from when this rule was first proposed, we vigorously opposed it. This is me testifying before that E&C committee. I was very honored. I was mentioning to Dr. Abruzzo this morning, I was actually sitting in the same chair that Anthony Fauci sat in many times when he was grilled by Congress. I was honored to sit in that chair. But there I was in front of this committee. And, you know, we basically, not only did I testify before this committee, but we had detailed written comments. We met with FDA leaders, with members of Congress, actually White House representatives. And at that hearing, the gist of my testimony was that we did favor legislation, and we felt valid, as imperfect as it was better than what the FDA was trying to do on their own, because it put guardrails around what the FDA could and couldn't do. We felt that full FDA oversight is necessary, but only for the very highest risk LDTs, which I will try to define for you in a minute. And that reopening CLIA legislatively is very dangerous. Many unintended consequences could flow from that, and we felt that that was not the right pathway to take. That final rule was released in 2024. FDA did not listen to a lot of the stakeholders, including the CAP. A lawsuit was filed within a few weeks by ACLA, the American Clinical Laboratory Association, then followed in August by AMP. And the CAP and other organizations filed amicus briefs in support of that lawsuit. An amicus brief is a way of a stakeholder basically supporting many of the contentions of the plaintiffs in a lawsuit, and so we filed a very strong amicus brief in favor of the lawsuits. And what we said there is that the FDA's action was arbitrary and capricious in violation of the Administrative Procedures Act, but more importantly, down at the bottom, it would put a huge burden on laboratories, the cost would be unsustainable, and it would impact patient access because these tests would not be available for patients. I'm happy to say that the federal court, as you well know, ruled in favor of the plaintiffs. And so this rule was vacated, fully vacated by the federal court. Now, the FDA could have appealed, and they chose not to appeal, and that deadline passed at the end of May. So this was our breaking news on the CAP website, that we were very happy that the deadline passed, that they would not defend this rule any longer, and that we applauded the judge's decision and the fact that FDA was not going to appeal this. Now, it's not over. There are continuing concerns about laboratory-developed tests, and what really still has to be decided, is this all going to be under CLIA by CMS, or all under the FDA, or both? I'll share with you maybe our current thinking as to maybe what we think might be workable. If the FDA is going to have any authority over LDTs, there needs to be new legislation because the rule vacated any authority that they would have to oversee LDTs. Oversight fully by CMS, we worry that many people feel that it would require a reopening of CLIA, and there are a lot of potential negative outcomes from that. Most stakeholders, certainly the CAP, and I think essentially all the stakeholders, and I've spoken with pretty much the leaders of every major organization that's in this fight, all believe that truly high-risk LDTs do need a higher level of oversight than the average LDTs that we develop in our laboratories. The question is, how do you define those? Here's a working definition that we currently have in the CAP. This is still evolving, but right now, these would be LDTs that produce a result that's not independently verifiable, and the consequences of an incorrect result would result in morbidity or mortality, and that some examples, tests that use unique, non-transparent, emphasis on non-transparent, proprietary technology, algorithms, and/or computations, such that the reported test result cannot be independently determined from the assay's result itself. In other words, you would need a confirmatory test. Or that interlaboratory comparisons cannot be performed, or PT, which is the main interlaboratory comparison, could not be performed. We estimate that tests that meet this definition are dramatically less than 1% of all LDTs. The non-transparency part here is really important, and a lot of what will ultimately be regulated will have to do with LDT and with AI and algorithms. And this sort of new concept of so-called explainable AI, where it's not fully non-transparent, is gaining momentum. And I think AI regulation is going to increasingly require that AI algorithms be explainable in some way and somewhat transparent. So, the CAP currently feels that maybe we need a balanced approach. Oversight of truly high-risk LDTs, we call them black box LDTs because you really don't know what's going on inside that box, do need a higher level of scrutiny, and we still think that the FDA probably is the best agency for that. Oversight of all other LDTs can safely be done by CMS, we feel, through CLIA. Now remember, that's 99% of all the LDTs that our labs develop and offer. We feel that any needed changes in CLIA should be done by rulemaking. Yes, no CLIA Act makes it a little bit of a new game, but we still feel that the rulemaking process, it would be much safer than just reopening this legislatively, because stakeholders would be trying to grab all kinds of advantages for themselves if it's totally reopened legislatively. We feel very strongly that whatever balanced approach is used, innovation must not be inhibited. That is pretty much number one, because that's where patient access really would suffer. So, why don't we turn to an even more pleasant topic, which is gene patents. You all know that this was a battle that was fought maybe 12, 13 years ago, and we thought we won, and we figured that it was always well. Well, nothing pretty much is always well forever when it comes to regulation. So, sort of important question around the whole myriad lawsuit and many other issues dealing with gene patents or patents in general is, what exactly is patentable? Did you know that we had the first patent law in this country was passed into law in 1790 and was signed by George Washington? That's how long we've actually been regulating and offering and issuing patents for things in this country. This was refined to a great deal by a patent law that was passed in 1952, so-called Section 101. People refer to it by that name. And it states the following: whoever invents or discovers any new and useful, number one, process, number two, machine, number three, manufacture, or number four, composition of matter, or any new and useful improvements thereof, may obtain a patent therefore, subject to the conditions and requirements of this rule. So those four categories of things are incredibly important, and the case law that surrounds patents pretty much revolves on those four things and or exceptions to those four things. So Congress has been keeping an eye on this for a very long time, and particularly recently. And this is from the Congressional Research Service, which, by the way, is a nonpartisan organization that does research for members of Congress. And they've been looking at this whole idea of patent eligibility. What can you patent? And as it says here, this is from their recent study, The U.S. patent system is designed to encourage innovation. It would be nice if we could stop there. The types of inventions that can be patented, however, may affect the patent system's ability to promote innovation in certain fields, especially in emerging technologies like artificial intelligence and biotechnology. So what they're saying is this still needs to be decided, what is really patentable or not, and Congress, arguably needs to look closely at these issues. So there's been a lot of legal precedents, literally all through the 1800s and all the way to the early 2000s. Several court cases, many of which were argued by the Supreme Court, created what are called judicially developed exceptions to the 4 categories of things that you can patent, inventions that you can patent. And here are the following 3 principles that have resulted from that case law. Things that are not patentable are laws of nature, natural phenomena, including products of nature. I should have underlined that because that was the whole hinge of the Myriad decision by the Supreme Court. And abstract ideas. I should underline that one too because that's where AI is going to reside. The whole legal concepts of abstract ideas, whether or not they even exist on a computer, there's an argument ongoing as to whether or not they can be patented. I'll give you an example of a case in just a minute. So this is a timeline, and I'm probably not readable from where you're sitting, but starting in 1900, adrenaline was patented, the discovery of adrenaline and the chemical structure of it. Patents were awarded for biological materials, but in the early days, not a lot of people were making very much money from that, so nobody really worried about it so much. Some important things happened in the 1980s. There was a case, Diamond versus Charbarty, if I'm pronouncing that correctly, that actually first ruled on whether or not a genetically modified microorganism could be patented, and it was ruled that it could be. Genentech went public in 1980, and the first recombinant DNA patent was issued, and a very important law, the Bayh-Dole Act, was passed and signed into law, which allowed not-for-profits and academic institutions to patent discoveries that were made using federally funded research. So many people in this room, I know at least one up here on the podium, had a patent that was patented, had a patent for a genetic discovery that was done using federally funded research. The first gene patents shortly after were issued to researchers at UCSF, and the Human Genome Project started, and it didn't take long before BRCA1, BRCA2 was discovered. And Myriad Genetics obtained the patents for that, and other people wanted to start doing work with BRCA1, BRCA2, and Myriad was stringent in their not allowing anybody to touch those genes. And that really affected a lot of progress, many of us believe. And ACLU and AMP filed a suit against Myriad. It was in the New York federal court. And by the way, you may not know that the federal court actually ruled in favor of Myriad at first. And only through the appeal process did it end up before this group. The Supreme Court, this was the Supreme Court in 2013, they agreed to hear the question, are human genes patentable? And they unanimously said no. And here's a quote from the majority opinion. A naturally occurring DNA segment is a product of nature and not patent eligible merely because it has been isolated. There was a lot of celebration when that ruling was made. I was directly involved in advocating on this issue back in those days, not for the CAP, but for the Association of Pathology Chairs, whose name has been changed recently. And so we were very happy. And interestingly, and here's the ruling in June of 2013, and there were actually a couple of other cases around that time, the Mayo versus Prometheus case and the Alice Corporation versus CLS Bank case. You've probably never heard of these until I was researching this talk, neither did I. But these were very important cases, and so the key recent cases in terms of this question include the AMP and Myriad Genetics case, which you know about, which dealt with isolated human DNA segments. The Mayo Collaboration Services versus Prometheus Labs dealt with whether or not you can patent a method for calibrating the dosage of a drug. The Prometheus Labs developed that. Mayo wanted to use it, and the Supreme Court ruled in favor of Mayo and against Prometheus Labs. You cannot patent that kind of methodology. Alice Corp versus CLS Bank was hinged on whether or not a method for mitigating settlement risk in financial transactions using a computer could be patented, and the Supreme Court said it cannot. There's that abstract idea issue. This is an abstract idea on a computer, and what is AI? But something very similar. So I think a lot of the discussion about AI and patentability is going to hinge on some of this case law. So I can remember back when a key argument that was made in favor of Myriad back in those days was that, if you change this patentability, that's going to turn the biotech industry upside down. R&D would come to a grinding halt. Subsequently, life-saving medications and technologies would never come to market. Well, looking back 12 years after that decision, it has been really just the opposite. I mean, innovation has exploded, and innovation dealing with genetic and other technologies and biomedical technology in general has literally exploded. So I think that argument today falls on deaf ears. But the issue has not gone away. The Patent Eligibility Restoration Act, and notice I give 4 dates for this because it's been introduced in Congress in 2022, 23, 24, and recently in 25. This was introduced by Senators Chris Coons and Tom Tillis as a way of establishing the ability to patent genes and genes association with disease. Now, you probably recognize that the states that are represented by these senators are extremely heavily populated by the biotech industry. Although we don't know that, we feel that industry has really been pushing hard to get back to the ability to patent genes. And so those judicially created exceptions would go away from those 3 key decisions that I mentioned. We are strongly opposed to this. The negative consequences, we think, would be tremendous. It would get us back to where we were, with Myriad and BRCA1, BRCA2. Loss of access to confirmatory testing, substantially increased cost of testing, even inhibition of gene-related research, because you'd have to get permission to do any research on a gene that's been patented. We're not alone in opposing this. You cannot read this list, but it's a pretty long list. CAP is in red there, but there are literally many, many, many dozens of organizations that are opposing this law. Here's sort of the main impact, we think, of being able to patent genes. Patenting a gene would limit the ability of developers to create tests and or therapies that would then deprive patients of those tests and therapies, and it would monopolize testing. Myriad had a monopoly. I don't know if you remember the issues, but Myriad was charging, I think, $4,000 or $5,000 for BRCA1, BRCA2. There are now BRCA1, BRCA2 assays that cost $250, and we would go back to that monopoly situation. Innovation, we think, would really be stifled. Gene-based research would be disincentivized. Developers would, not be in a position maybe to develop build on new gene discoveries. And so it would just stifle a lot of the progress that we've enjoyed over the last 13 years or 12 years. And it would even impact healthcare equity. As cost would go up, it would make inequity even greater, and manufacturers might only focus on the things that can make the most money and not necessarily help the most people. So I have tried in 35 minutes to go over a lot of material with you. And again, I promise you already knew all of this. I think I was right. And hopefully I've given you a little food for thought as to how we need to approach this regulation and how we need to really work closely with agencies such as the FDA. I will be sitting down with the new FDA commissioner in 4 weeks. with a group from the CAP to try to convince him how we feel this should be safely regulated going forward. So I think we do have a few minutes for questions, so thanks very much for your attention.
Yes, Yasmin, go ahead.
Thank you so much. This was fantastic. We really, really enjoyed it, and thank
Great.
you for being here.
Great.
I would like to beg your forgiveness for bringing up a topic that's probably really dear to this group's heart, but it has been unresolved for a long time, which is PhD billing. This is a crowd that actually is mostly PhDs that are doing interpretation and doing really the testing that patients need. Where do we go from here? What is CAP's advice for us? How do we make it work to bring, speaking of equity, to really bring equity to the work that we do?
So that's a great question, and you may be surprised to know that the CAP strongly supported PhD billing for molecular analysis in 2010, 2011, even into 12, when CMS was deciding what fee schedule should molecular analysis be on. Should it be on the physician fee schedule, or should it be on the clinical laboratory fee schedule? I'm talking about Medicare patients, but as you know, what happens in Medicare pretty much happens in the rest of the laboratory or the rest of the healthcare landscape. CMS ruled against it. They said that PhDs should not be allowed to bill for these services, and also these tests were put on the clinical laboratory fee schedule. There are very limited, as you know, very limited codes for physicians to bill for these services. So where we are today, we have talked with CMS about this issue, and they have remained adamantly opposed to it. And so, you know, we are open to figuring out a way for this to work, but CMS has continued to be consistently opposed to it. I don't know if that helps you knowing that, but I mean, we were strongly in favor of it, and we felt that it would all fit together nicely in 2011-ish for a fee schedule where these tests would be the people that run and interpret these tests could be adequately rewarded, and we included PhDs in that.
Pardon my ignorance here. I'm used to receiving proficiency testing results where it lists the people who have contributed towards the testing system. Is there a place where we can see the contributing authors and regulators for our CAP guidelines that we follow in the molecular pathology checklist?
Yeah, absolutely. I mean, these things are developed by the various committees of the CAP. There are 82 committees in the CAP, believe it or not, of which 31 or 32 are our scientific committees, and they're the ones who actually develop the PT products. I mean, I couldn't give you, I don't know that I could even find out which author was behind which PT product that was developed, but you can certainly see by just looking at the rosters of those committees, the kinds of people that are working to develop these products. The checklists, we have multiple committees, including a checklist committee in our Council on Accreditation, and the names of those folks are listed there as well. We try as best we can to make sure that the right people are on those committees. And by the way, many of those scientific committees, I hope you know this, are populated by non-CAP members, by PhDs, because we need the input and expertise of the PhD community on most of those committees. So I don't know if this is helping you.
Yeah, it does, because my next thing I'm going to ask, putting the CGC president hat on my head, is there a role for cross-fertilization, having CGC members and representatives in those, so that there's AMP maybe members?
Absolutely.
Is there a way that we can contribute to that, given that we are at the bench? We're not doing clinical interpretations, we're doing that work.
Absolutely, and I standing here, I can't tell you if we have any CGC members on any of the committees. If someone in the room is on a CEP committee,
But I mean, recognize.
I don't see any hands going up, but we have, oh, we do have.
But recognized as a CGC member.
Yeah, many of the representatives that are non-CAP members are representing different organizations. And so I confess that I didn't know that one person in this room, CGC member, is on one of our committees. But we can absolutely make sure that we include folks from this organization in future assignments on those committees. It's essential. We cannot, I mean, you know, we pathologists, we don't have the skill set and the background knowledge to develop a lot of these products, and we really rely on the non-MD, the PhD community.
Thank you. My second question is more of a personal one coming from experience about being a CAP reviewer, right, a surveyor, is that given that, as you pointed out, that the molecular pathology checklist is getting longer and longer, and perhaps that is a good way for, you know, surveying more labs to standardize. Our list is getting longer, but the times of the surveys are getting less. So, you know, oftentimes there's a crunch as to you can't get into each and every checklist item. Do you have any recommendations that perhaps some of that with the expanding list can be put a little side or extended in terms of time?
So we are always trying to simplify our checklist. I know that may not feel like it when you're actually going to do an inspection. However, you probably will not be surprised to hear that we are looking at AI tools to help inspection team members and team leaders do a lot of the database analysis before you actually even show up in the lab to do the in-person inspection. And it hasn't been rolled out yet, but we're actually looking at a way of so you won't spend 4 hours reviewing documentation in the morning. We would rather the inspectors be out at the bench talking with techs and finding, you know, witnessing that things are being done the way they're supposed to be done.
Thank you.
So stay tuned. It's
Thank
coming.
you.
Okay.
I think we have time for just one more question.
I just want to make a comment, Jane. There are a number of CGC members who, from both the CAP side and the ACMG side, who have been on CAP committees, the cytogenetic committee, molecular genetics, molecular oncology.
Yes.
So, I mean, I think in that respect, CGC has been represented. However, there are also liaisons from different organizations to CAP committees, and maybe that's a possibility.
Absolutely. We already have those, as you noted, and we actually have outbound liaisons to many other organizations, and I need to investigate how we can maybe have bi-directional liaisons with CGC and CAP. And I think there are no more questions.
That was wonderful. Thank you so much. So we now have a coffee break. Please remember to visit our exhibitor booths. And the Science Cafe presentations are by BioNano, BioView, and Illumina. And thank you, Dr. Karcher.
You're very welcome.